Hello everyone, welcome to another MCAT ACE lecture for the biology and biochemistry section. In today's lecture, we are going to describe different metabolic pathways that are required for energy production. I'm going to split this lecture into two parts. Part 1, which will cover glycolysis, fermentation, citric acid cycle, and electron transport chain. And then in the second part, we will cover the rest of the material that are listed here. And then as always, at the end of the lecture too, we will go over some practice test questions. Alright, so now let's start with glycolysis. Here I'm showing you the fate of the glucose, the main pathways that it will have to go through. And if you know these pathways, you know this lecture pretty well. So for the first part of this lecture, we are going to cover glycolysis, how glucose will be broken down into pyruvate, and then depending on the availability of oxygen, pyruvate can undergo aerobic respiration through the tricyclic acid or citric acid or Krebs cycle. All are the same names. And then from there on, its products will be sent to the electron transport chain to produce ATP. And then if there is not enough oxygen, then the pyruvate will have to undergo anaerobic respiration or fermentation. And we will cover all these reactions in details. And then for the second part of the lecture, we will discuss how glucose can be stored in the form of glycogen, starch, sucrose, or it could undergo the pentose phosphate pathway, also known as hexose monophosphate shunt, in order to produce ribose 5-phosphate and NADPH. And again, we will go over all these pathways in detail, so don't worry about it if there is something that you're not sure about what's going on here. Alright, so starting with glycolysis, its definition is actually breakdown of glucose. And what happens in glycolysis is that glucose through a series of reactions will be broken down into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, or DHAP, dihydroxyacetone phosphate. And these two molecules are isomers and are interchangeable, so therefore we can say that the glucose will give the final product of two molecules of glyceraldehyde 3-phosphate. And so here, let me erase that for you. So there we go. One glucose molecule that is made of 6 carbon is broken down into 2 molecules of glyceraldehyde 3-phosphate, each of which is a 3-carbon molecule. So 2 3 carbons will add up to 6 carbon here. And from here on, glyceraldehyde 3-phosphate will go through a series of reactions that will yield pyruvate. Alright, now we can split these reactions into two parts. One is the preparatory phase. So here the first section is called preparatory phase, and the reason for that is that these first few reactions will require 2 molecules of ATP, 2 ATP, and then it will give out 2 ADP. And so it's consuming energy in order to get the reaction going. And then in the second phase of the reaction, which is called the payoff phase, each molecule of glyceraldehyde triphosphate will give 2 ATP and 1 NADH. So here we will also have 2 ATP and 1 NADH. And now if you look at all these reactions, 2 ATP was consumed in order to yield 4 ATP and 2 NADH. So we can say that the net reaction of glycolysis is glucose breakdown by adding 2 ATP and 2 NAD plus and 2 phosphate groups to yield 2 pyruvate plus 2 ATP, 2 NADH, 2H plus and 2H2O. So here 4 ATP minus 2 ATP that was initially consumed yields 2 net ATP. And then 1 NADH and another NADH for the second glycerol triphosphate will yield 2 NADH. So this is basically the net reaction that you should be familiar with for the glycolysis. Okay, now that you're familiar with all these reactions, we can move on to describing in details every one of these reactions. And so here I've written the pathway for you and then the fate of the pyruvate in the end. But here it could be a little confusing because I have the rest of the pathway. So what I would like you to do is just to focus on the glycolysis reactions here. Just forget the rest of the stuff that is going on here. And then later on we will cover all the other pathways. So here, let me clear the page for you. And there we go. Now we have a new page covering the glycolysis reaction. Now, in order to remember all these different intermediates that we're going through, I have my own memory aid that you use. If you also created your own memory aid that you feel comfortable with, then by all means you can go ahead and use that one. But the one that I like to use myself is grapefruit has a good amount of phosphate. And so what it stands for is G is for 2 first glucose molecules, fruit is for 2 fructose, so fructose 6-phosphate, and then we have fructose 1 and 6 bisphosphonate. And then G stands for glyceraldehyde triphosphate, and then D stands for diphosphoglycerate, and it's the same name as biphosphoglycerate. So it depends on which book and which resource you're using. Some are saying 1 and 3 biphosphoglycerate, the others are saying 1 and 3 diphosphoglycerate. They're all the same thing. And then finally, for the phosphate group, we have 3 different phosphates to list here, which includes 3-phosphoglycerate, 2-phosphoglycerate, and then phosphoenol pyruvate, and then finally the final product is, we all know, is pyruvate. Okay, so now we can go in and fill in the gaps. The way again I do it is that I say that we have to give an ATP in the middle of glucose and an ATP in the middle of fructose, and then in between the good OO right here we're gonna get an NADH back, and then we also get ATP around the phosphate. So we get an ATP out in between the reaction between diphosphoglycerate converting into 3-phosphoglycerate, and then from phosphoenol pyruvate converting to pyruvate, we're also gonna get another ATP molecule. So basically here now you have all the substrates and the intermediate molecules and then their products of different ATP and NADH that they would yield using the grapefruit has a good amount of phosphate. Alright, now let's go in a little more detail regarding every one of these molecules. And now I just want to give you another mnemonic that some students find helpful, is that about the structure of these molecules. Sometimes they ask, so how do we remember the structure? They're too confusing and too many, and I agree. So that's why I always use memory aids to help myself remember these. So what I do is that for the first one, glucose, glucose, fructose, fructose, I remember that they're like a circle structures. And then after that, for glyceraldehyde triphosphate and dihydroacetone phosphate, I remember them as EE. And then for the rest of these reactions, I remember their shape as M, 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 and then phosphoenol pyruvate and pyruvate, I remember them as resembling like an H. Okay, now with that information in mind, let's go and draw every one of these reactions. So first we have the glucose here, and we all know that glucose is a 6-membered ring. Alright, we have an OH, OH, and an OH group. So here we've got our first molecule here, which is glucose. Via the hexokinase enzyme, it will replace the OH group with a phosphate group. So here, let me erase the OH group and show you that we have an O phosphate, double bond O, O negative, and then O negative. And so now we have a glucose 6-phosphate molecule. So here, let's count the carbons. We have carbon 1, 2, 3, 4, 5, and 6, and the phosphate group is located on carbon 6, so that's why we call it glucose 6-phosphate. And recall, I just mentioned that their structure is a circle, so we are still in the ring structure for the second molecules. The next two are also going to be ring, and so that's how the memory aid helps you remember the structure of all these molecules. And then referring back to the grapefruit has a good amount of phosphate. You remember that the first reaction, glucose conversion into glucose 6-phosphate, required an ATP molecule. So here you can see that an ATP molecule has gone in, consumed, and an ATP was given out. And the phosphate from adenine triphosphate is used for phosphorylation of glucose. So here, one less phosphate here. The phosphate is added to the 6th carbon, as a consequence of which we now have adenine diphosphate. Now the next reaction, via the isomerase enzyme, glucose 6-phosphate will be converted into fructose 6-phosphate, which is still in the ring structure, but it's a 5-membered ring. So let's draw the structure of fructose 6-phosphate now. OH, and then here we have the phosphate group. So instead of drawing all the O's and P's and so on, and for simplicity I'm just going to show a circle with a P inside for the phosphate group. Right now we have fructose 6-phosphate in this group. The next reaction is conversion of fructose 6-phosphate into fructose 1 and 6-bisphosphonate, and this is being carried out by the phosphofructokinase 1 enzyme, which adds a phosphate group to carbon 1 of this group, and this is the rate limiting enzyme of the glycolysis pathway. And again, referring to the memory aid I had for you, you know that conversion of fructose 6-phosphate into fructose 1 and 6-bisphosphonate requires another molecule of ATP in order to add a phosphor group. And so here for carbon 1 here, let's eliminate the OH and replace it with a phosphate group. Alright, so now here we have fructose 1 and 6-bisphosphonate. With the help of aldolase enzyme, fructose 1 and 6-bisphosphonate will be converted into glyceraldehyde triphosphate and dihydroxyacetone phosphate. And remember I said these structure has like the E shape, so here let's draw them. First we have the glyceraldehyde triphosphate and dihydroacetone phosphate. So here we have an OH group, double bond O, and a phosphate. That's for dihydroacetone phosphate. And then for glyceraldehyde triphosphate, we have a double bond O, OH. So the place of these two molecules is changed, and then we have a phosphate group. And these two molecules are isomers and are interchangeable. So therefore, we can here say that now one glucose molecule, which was made of 6 carbon, has yielded 2 molecules of glyceraldehyde triphosphate, which is a 3 carbon molecule. So from here on, that's why I'm showing you 2 arrows for every reaction, because we have 2 molecules of every one of these products that we are going through. The next reaction is conversion of glyceraldehyde to the different phosphate groups. That's how I remember. Remember, grape fruit has a good amount of phosphate. And so here we have the glyceraldehyde converting into diphosphoglycerate, and if you recall, it was giving an NADH out here, and then before the phosphate we get an ATP out, and after the phosphate we get an ATP out. So here we have the glyceraldehyde triphosphate converting into 1 and 3 diphosphoglycerate, and it has an M shaped from here on. So I'd like you to remember all those memory aids. Just have to erase them because I need more space to draw them. But here, now we have an M shape diphosphoglycerate. It has 1 and 3 diphosphoglycerate. Alright, just need a little more space. Alright, we have the O, then we have the phosphate group attached to the first carbon here. Second carbon has an OH, and then the third carbon also have another phosphate group. So here we have 1 and 3 diphosphoglycerate. That was for D. Now, from here on, we will have different phosphate group. And the way I'm calling it different phosphate group is that we have 3 phosphoglycerate, 2 phosphoglycerate, and then phosphoenolpyruvate. So now let's list them out here. Via the phosphoglycerate kinase enzyme, the 1 and 3 DPG will be converted into 3 phosphoglycerate. And so it means that now only the third carbon is holding the phosphate group, and the first one has lost it. So here, let's erase that one, the phosphate here, and replace it with an O negative. So now we have 3 phosphoglycerate. And then through the next reaction, we are getting via the phosphoglycerate mutase, converting the 3 phosphate group into the 2 phosphate. So now let's move the phosphate group from the third carbon to the second carbon. All right, and if you pay attention, all these structures still have that M configuration that I told you about. Alright, now the second carbon has a phosphate group, and here we have an OH, O and O negative. So now we have a molecule of 2 phosphoglycerate. That's where we are in our pathway. And then with the help of the enolase enzyme, it will be converted into phosphoenolpyruvate, which from here on they have an H shape. And so let me clear out the board for you and draw it again. Here we have a phosphate, double bond O and O negative. So here we have phosphoenolpyruvate, and then through the final reaction, pyruvate kinase will convert phosphoenolpyruvate into pyruvate. So here again we have another H structure, double bond O, OH, and double bond O, and that's about it. We're done with our reaction here. So now we have the pyruvate. Okay, so if you can grab a piece of paper and try to draw all these reactions for yourself, after doing it 2 or 3 times, you just realize that it's not too hard and you remember all of them. But the main point I'd like you to remember is the memory aid that I provided here. Grape fruit has a good amount of phosphate. Then the final product was pyruvate. Okay, so here we had glucose converting into glucose 6-phosphate. Then we had fructose 6-phosphate converting into fructose 1 and 6-bisphosphonate. Then we had glyceraldehyde 3-phosphate converting into 1 and 3 diphosphoglycerate. Then we had all the phosphate groups here, which included 3 phosphoglycerate, 2 phosphoglycerate, phosphoenolpyruvate, and then the pyruvate. Now the question that I had here for you is that which of these reactions required ATP? In between the glucose and fructose, remember, they needed the ATP. And so that's why these were the preparatory phases, because they consume energy. And then which reactions were giving out ATP? In between the phosphate. So conversion of 1 and 3 diphosphoglycerate to 3 phosphoglycerate and phosphoenolpyruvate to pyruvate. These reactions will give ATP. And how many ATP do they give out? From here on, everything is times 2 because we have 2 molecules of GTP. So 2 ATP here and 2 ATP there. And then the final question that I have for you is that which of these reactions will give out an NADH molecule? Here, these 2 eyes are looking at you right there. Good. So right here, it will give you an NADH molecule, but then we have 2 molecules of glyceraldehyde triphosphate. So 2 NADH will be given out here. Alright, and that's about it. That was the summary of the glyceraldehyde pathway. Now I'm going to go back to the original pathway that I'd shown you on the screen. So if you just give me a minute, I'm going to clear the board here. Alright, there we go. We are back now with all the reactions. If you need to pause the video and rewrite these things, then go ahead by all means. But then here is the net reaction of the glycolysis pathway. It describes how glucose has been converted into 2 molecules of pyruvate. And now from here on, depending on the availability of oxygen, the fate of the pyruvate will be determined. If there is enough oxygen in aerobic organisms, pyruvate can undergo aerobic oxidation, where conversion of pyruvate into acetyl coenzyme A will cause a release of carbon dioxide and generation of an additional NADH molecule per reaction. So since we have 2 molecules of pyruvate, therefore we will get 2 CO2 and 2 NADH. And now the product, acetyl coenzyme A, will enter the citric acid cycle. So how many carbon is the acetyl coenzyme A? So remember, pyruvate was a 3 carbon molecule, but we have lost the CO2 here. So this acetyl coenzyme A is a 2 carbon molecule. It will enter the citric acid cycle, which the starting molecule here is the oxaloacetate, which is a 4 carbon molecule. But then once it combines with a 2 carbon molecule, it yields citrate, which is a 6 carbon. And throughout this whole reaction, 2 CO2 molecules will be released, 3 NADH, 1 FADH2, and 1 ATP will be produced. But then since everything here is 2 molecules that we are going through, we have to multiply all these numbers by 2. Alright, so we will later on discuss the citric acid cycle in a little bit in just more details, but for now let's focus on the fate of pyruvate. So if we had the oxygen available in aerobic organisms, pyruvate can undergo aerobic respiration, produce more NADH and FADH2, and these products can then later on go through the electron transport chain to produce more ATP. But if there is not enough oxygen, then pyruvate has to undergo fermentation. And the purpose of fermentation is to produce more NAD+. So if you've noticed, all the NAD+ that we had here is being used up in the glycolysis pathway. And if we don't regenerate the NAD+, the anaerobic organisms, or under hypoxic conditions, our cells cannot generate more ATP because they need NAD+ in order to get the glycolysis go through. And so that's the whole purpose of fermentation, that is to regenerate the NAD+ that was used up during the glycolysis reaction. And so we have 2 types of fermentation, like lactic acid fermentation seen in animals and bacteria, or ethanol fermentation, which is seen in yeast and some bacteria. And we will go in more details regarding these reactions again. One last point that I would like to mention about glycolysis before I move on to the citric acid cycle is regulation of glycolysis. Recall I mentioned that the phosphofructokinase, this is the rate limiting step of this reaction. So anything that controls the rate of this enzyme is basically controlling the rate of the glycolysis right here. And allosteric molecules that are having positive effects are ADP and AMP, and the allosteric molecules that have negative effect, meaning they would suppress these enzymes, are ATP and citrate. And you will see we will do many more examples of how different hormones or different allosteric molecules affect the rate limiting enzymes. But instead of memorizing all these molecules, I want you to know the concept. So what's the purpose of glycolysis? Its purpose is to produce more ATP and also increase acetyl coenzyme A production so that it would undergo the citric acid cycle and so produce more ATP through the aerobic respiration. So therefore, if we have high level of ATP or high level of citrate, it means that we've already got enough energy going on. There is no point for glycolysis to keep going. And so that's why these two molecules, ATP and citrate, have a negative influence on phosphofructokinase. They would downregulate it, so glycolysis rate will slow down, so we don't get as much ATP and citrate anymore. On the other hand, if we have too much ADP and AMP, it means we don't have enough energy there. We need ATP for the cell, the energy source. And so if there is too much high concentration of ADP and AMP, there would be positive feedback on glycolysis reaction via activation of the phosphofructokinase in order to produce more ATP. So in other words, instead of memorizing what allosteric molecules have what effects on different rate limiting enzymes, I want you to know the concepts, and then during the exam you can figure it out. Just think through it and find out, all right, what's the purpose of these molecules and what's the purpose of the reaction. And so you can easily find out which direction different allosteric molecules will direct different pathways. Now moving on to one other concept is the glycolysis feeder pathways. So we already know that glucose was the starting molecule, but then there are other molecules that can be added to this glycolysis pathway in order to get the glycolysis going. And so I'm going to discuss about all these intermediate molecules in the glycolysis feeder pathway. So there are 4 different groups of molecules that can contribute to the glycolysis. These include glycogen and starch, disaccharides, and monosaccharides. All right, now let's go through every one of them. Glycogen is the storage form of glucose, where we have a chain of glucose with some branches that are available. And so once we break down these glycogen molecules into smaller glucose molecules, now glucose can enter glycolysis pathway and contribute to production of energy. So I'm going to discuss the glycogen reactions at a later time during this lecture, but for now I just want to focus on the concept of which molecules can contribute to the glycolysis. So glycogen is the first one, where conversion of glycogen 1-phosphate via the phosphoglucomutase. Again, don't worry about the name, I'm gonna cover them all separately, but conversion of glucose 1-phosphate via the phosphoglucomutase into glucose 6-phosphate will give us an intermediate molecule that can now enter the glycolysis pathway. So there we go. Glycogen breakdown gave us glucose 1-phosphate, which has been now converted to glucose 6-phosphate and can now resume the glycolysis. The next molecule is starch, which is again made of a bunch of glucose molecules that are bound together. This here we have the 1 and 4 glycosidic linkage. So again, here we have carbon 1, 2, 3, 4, 5, and 6. And so here we have the 1 and 4 glycosidic linkage. Via the amylase enzyme, starch will be broken down into a disaccharide called maltose, and then the maltase enzyme will break down maltose into the glucose molecules, and now glucose can undergo glycolysis. Moving on to disaccharides, which includes the maltose, that is made of 2 glucose molecules that are bound together, lactose, which is made of a glucose and a galactose molecule that are bound together, and lactose is found in milk. And then we have sucrose, which is made of a glucose and a fructose molecule. Now breakdown of all these disaccharides, like breakdown of maltose by maltase, will give us glucose that can now enter the glycolysis pathway. Lactose breakdown will give us glucose. Again, glucose, all these can enter the glycolysis pathway. And then the other sugar, like galactose and fructose, via a series of reactions, these molecules can also enter the glycolysis pathway. So let's cover them. With galactose, the galactokinase enzyme will yield galactose 1-phosphate, and then this molecule via uridyl transferase will be converted into glucose 1-phosphate, which can now enter glycolysis or used for gluconeogenesis or any other purposes. And then with fructose, there are two different enzymes that can help convert fructose into molecules that can resume glycolysis. The first one is hexokinase, which is found in kidney and muscles, and hexokinase converts fructose into fructose 6-phosphate. And fructose 6-phosphate is already one of the intermediates of the glycolysis pathway. So there we go. We have fructose 6-phosphate here. And then the other enzyme that we got is called fructokinase. Fructokinase, which converts fructose into fructose 1-phosphate. And then from here on, the aldolase enzyme converts fructose 1-phosphate into either dihydroacetone phosphate or glyceraldehyde triphosphate, which now are part of the glycolysis intermediate molecules. And then finally we have the monosaccharides like mannose, which is another hexose sugar that is closely related to glucose and is often released from digestion of different polysaccharides and glycoproteins. Now mannose via the hexokinase enzyme will be converted into mannose 6-phosphate, which via the isomerase converts into fructose 6-phosphate, and then fructose 6-phosphate can now enter the glycolysis reaction and carry out this pathway. Alright, so basically that's about it about glycolysis. Now we can move on to the aerobic respiration from here on. So far we have described the glycolysis, explaining how glucose is converted into pyruvate. Now I'd like to describe the next pathway, which is the aerobic oxidation. Once we are done with that one, then we will proceed with the other pathway, which is anaerobic reduction or fermentation. Alright, now here we have the citric acid cycle. So basically under aerobic conditions, and of course it has to be the aerobic organisms, pyruvate undergoes oxidative decarboxylation by the pyruvate dehydrogenase enzyme. So here we have pyruvate. It's a 3-carbon molecule. With the help of the pyruvate dehydrogenase enzyme, a CO2 molecule will be lost and an NADH molecule will be produced. And remember, a glucose yielded 2 pyruvate, so therefore everything is multiplied by 2 if we want to know a fate of one glucose molecule that we are discussing here. Now the product of this decarboxylation is acetyl coenzyme A, which is a 2-carbon molecule, and now this acetyl coenzyme A can enter the citric acid cycle. And so here we have the oxaloacetate, which is a 4-carbon molecule. Once it's added to a 2-carbon molecule, it forms citrate, which is now a 6-carbon molecule. And then through a series of reactions that goes through here, the 2CO2 will be released. So the cycle repeats because we got 2 carbon here, but then 2 CO2 are released, so the 4 carbon oxaloacetate will be regenerated eventually. 3 NADH will be produced, 1 FADH2 will be produced, and 1 GTP or ATP will be produced. And again, we have 2 molecules of everything here. So 2 times every one of these values that we just discussed. And that was basically a brief summary of the citric acid cycle, also known as Krebs cycle, or the other name is tricyclic acid cycle. That's why they say TCA cycle. Right. Next, I'd like to now go over all the reactions that are going on here in the citric acid cycle. And for that, again, I have a memory aid that I hope you'll find helpful. All right, so here is our memory aid. If you can memorize the sentence, then you can memorize all the intermediate molecules that are going through the citric acid cycle. Our citrate city is kept safe and sound from malaise. All right, so we have the citric acid cycle. R stands for oxaloacetate. Citrate stands for citrate, that's easy. City stands for cis-aconitate. Is stands for isocitrate. Kept stands for alpha ketoglutarate. Safe stands for succinyl coenzyme A. Sound stands for succinate. From stands for fumarate. And then malaise stands for the mullet. Alright, so now you know all the different molecules involved in the citric acid cycle. You should also know which of these reactions are releasing NADH, FADH, or ATP. So let me show you again. We had pyruvate here that yielded acetyl coenzyme A, and now it has entered the citric acid cycle. So before entering the citric acid cycle, we got an NADH right here and a CO2. After entering the citric acid cycle, these 2 reactions right here will yield an NADH and a CO2. NADH and a CO2. And then conversion of succinyl coenzyme A into succinate will convert a GDP into GTP, so GDP into GTP, and then GTP can be used to convert ADP into ATP. So that's why you see some of the sources say citric acid cycles, 1 molecule of glucose gives 2 ATP, some other ones say 2 GTP. It's because they're interchangeable, so you can get ATP back from GTP. And then fumarate conversion, succinate conversion to fumarate will give you an FADH2, and the way I memorize it is that fumarate requires FADH2. And then in the final reaction, conversion of malate into oxaloacetate will give you an NADH. So now you have all the molecules, all their products. The last step is to find out which enzymes is required for every one of these reactions. And so if you give me a second, I'm going to clear the board and bring up a new page to review the enzymes for the citric acid cycle. All right, there we go. Now here we have the cycle. Now I'd like to review different enzymes for this cycle. So we've already discussed the first reaction. This is carried out by the pyruvate dehydrogenase. Enzyme. Then addition of acetyl coenzyme A to oxaloacetate is being done by the citrate synthase. We have to generate citrates, so it's being done by citrate synthase. That's pretty easy. The next two reactions is being done by aconitase. Conversion of isocitrate into alpha ketoglutarate is being done by isocitrate dehydrogenase. Conversion of alpha ketoglutarate into succinyl coenzyme A is being carried out by alpha ketoglutarate dehydrogenase. Conversion of succinyl coenzyme A to succinate is being done by succinyl coenzyme A synthase. Then conversion of succinate into fumarate is being done by succinate dehydrogenase. Fumarate into L-malate is being done by fumarase. And then malate into oxaloacetate is being done by malate dehydrogenase. All right. And so these were all the reactions that were involved. Now next I'd like to go over the most important reactions that are involved in controlling the rate of the citric acid cycle. So there we go. Now we are back to the first page. Let's go over the different reactions that control the rate of the citric acid cycle. So basically the overall rate of this pathway is controlled by 1, the rate of the conversion of pyruvate into acetyl coenzyme A, and then second, by 3 of the enzymes that are in the citric acid cycle. And if you look at different sources, some of them say, oh, isocitrate is the rate limiting step of citric acid cycle. Another book may say, oh no, it's the alpha ketoglutarate. And so basically, after reviewing multiple different sources, the conclusion that I came to is that all these 3 enzymes, citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase, all 3 are the rate limiting enzymes, except that it's under different circumstances that one of them may be more important than the other one. All right, so these 3 enzymes control the rate of citric acid cycle, and also pyruvate dehydrogenase is another determinant of the rate, because citric acid cycle is dependent on the production of acetyl coenzyme A. And so first let's talk about these 3 reactions here. Again, we have to know the concept again. I don't want you to memorize, all right, what are the allosteric inhibitors or activators for all these enzymes. But then if we have high products, high concentration of the products of the citric acid cycle, then this cycle needs to be inhibited. And these products include, for instance, ATP and NADH. If you have high concentration of these molecules, then the activity of these enzymes will be decreased. Likewise, for pyruvate dehydrogenase, if we have high ATP, high NADH, or high acetyl coenzyme A, or high fatty acids, because acetyl coenzyme A can be converted to fatty acids, then again the rate of pyruvate dehydrogenase will be decreased. And then on the other hand, if we have high concentration of ADP, NAD+, or AMP, then it means that we don't have enough energy, so the rate of these reactions will increase in order for citric acid cycle to produce more energy. Same for pyruvate dehydrogenase. If you have AMP or NAD+, or high concentration of coenzyme A that needs to be converted into acetyl coenzyme A, then the rate of the pyruvate dehydrogenase will increase. And then the final molecule I'd like to discuss here is calcium. This molecule also helps regulate the citric acid cycle by activating the pyruvate dehydrogenase. isocitrate dehydrogenase, and alpha-ketoglutarate. And one reason for this activation is that in muscles, once the calcium concentration increases, in order for muscle to contract, we also need more energy. And so by activating these enzymes, there would be increased ATP production and aerobic respiration. Alright, so now we are done with the citric acid cycle. Next, we can move on to the electron transport chain and explain how different one of these reactions, how different products of the citric acid cycle and glycolysis in the electron transport chain can contribute to the production of ATP. So there we go. The glycolysis and the citric acid cycle, they had contributed to producing NADH and FADH2 molecules that will attribute to production of ATP via the electron transport chain. So any ATP that has been produced during the glycolysis reaction or during the citric acid cycle is called substrate level phosphorylation. But then all the products like NADH or FADH2 that will undergo oxidative phosphorylation in the mitochondria, any ATP that are produced from those molecules is called oxidative phosphorylation. And now you can see that I'm showing glycolysis outside the mitochondria. It's because glycolysis is being carried out in the cytoplasm, while the citric acid cycle happens in the mitochondrial matrix, and then the oxidative phosphorylation is carried out in the inner mitochondrial membrane. Alright, now before I go over the structure of mitochondria with you, I'd like to remind you of all the products that we got from glycolysis and the citric acid cycle. So remind me again, what products did we get from glycolysis? We got 2 ATP and 2 NADH. So we consumed 2 ATP but yielded 4 ATP out during the glycolysis. That's why we say we have a yield of 2 ATP and 2 NADH. And then glycolysis yielded 2 pyruvate molecule. Conversion of pyruvate into acetyl coenzyme A also gave us 2 NADH. And then the citric acid cycle itself gave us 3 NADH, 1 FADH2, and then 1 ATP. But since we had 2 molecules of acetyl coenzyme A, so everything will be multiplied by 2. So overall, all these reactions of glycolysis and citric acid cycle gave us 4 ATP, 10 NADH, so here 6 NADH right here, and then another 4 NADH there is giving us 10 NADH, and 2 FADH2. So these 4 ATP are produced via substrate level phosphorylation. Now these NADH and FADH2 will undergo oxidative phosphorylation in the electron transport chain to produce ATP, and this is called oxidative phosphorylation generation of ATP. Alright, so that was a nice review of all the reactions that we've been discussing since the beginning of this lecture. Now I'd like to go over the mechanism of the electron transport chain. So mitochondria contains an outer membrane that is permeable to most small molecules and ions, and an inner membrane that is impermeable nearly to all ions and uncharged molecules. Now the inner membrane is forming folds called crista, or if you want to add them up, it's called cristae, and some people call them cristae. And so the purpose of all these folds or cristae is to increase the surface area of the mitochondria, because imagine we didn't have any of these folds. That would be our inner membrane. How much space do we have? Not that much. Versus look at all the space that now we got here from all these folds here. So it increases the surface area required for the electron transport chain. Now this inner membrane is what contains the electron transport chain as well as the ATP synthase enzyme. The space between the inner membrane and the outer membrane, so the space right here, this is called the intermembrane membrane space, and the space bounded by the inner membrane, the space right here, is called the mitochondrial matrix. And this mitochondrial matrix, as we just discussed, contains all the enzymes required for the citric acid cycle and the fatty acid oxidation. Right, so now here I have a drawing of the electron transport chain for you. Here we have the outer mitochondrial membrane, the inner mitochondrial membrane. The space between the two is called the intermembrane space, and the space bounded by the inner membrane is called mitochondrial matrix. Let me clear the board here for you and provide some background information about oxidation reduction reactions that is going on here in the electron transport chain. So the chemical reactions in which electrons are transferred from one molecule to another, these are called oxidation reduction reactions, or you can say redox reactions. Alright, now during this process, the electron donating groups are called the reducing agent. And then the electron accepting, so this was electron donating group. And then the electron accepting group is called an oxidizing agent. And so here we can see that NADH and FADH2, they start as reducing agent, transferring electron via the complex 1 and complex 2 of the electron transport chain into the ubiquinone molecule. Now next, ubiquinone has become a reducing agent, and via complex 3 it transfers electron into cytochrome C. Now cytochrome C is a reducing agent and will transfer electron to the oxygen. And throughout all these reactions, what happens is that the energy is released from electron transfer in every one of these reactions, and this energy that is released is used to pump proton or H plus from the mitochondrial matrix into the inner membrane space. So you can see that by complex 1, H plus is pumped out, by complex 3, H plus is pumped out, and then by complex 4, H plus is pumped out. So one NADH molecule has pumped out 3 H plus molecules. And so that's why that we always say that NADH yields approximately 3 ATP molecules through the electron transport chain. Now I'd like you to pay attention here. FADH2 on the other hand, it doesn't go through the complex 1. It goes through complex 2, and complex 2 cannot pump any hydrogen. So unlike NADH, FADH2 only pumps 2 hydrogen from the matrix to the intermembrane space. And so therefore, every FADH2 molecule will yield approximately 2 ATP in the electron transport chain. And then the energy from the electrochemical gradient of the protons here in the intermembrane space will be used to charge the ATP synthase enzyme in order to produce ATP. And I will go through more details regarding the function of the ATP synthase. For now, I'd like to focus a little more on these complexes and discuss their enzymatic activity. Alright, so we have complex 1, 2, and 3 and 4 that I'd like to review. So complex 1 is made of NADH dehydrogenase, which catalyzes oxidation of NADH by coenzyme Q. So here NADH was a reducing agent. It's undergoing oxidation by coenzyme Q, and so now we have a reduced form of coenzyme Q. And this coenzyme Q later on via the complex 3 will help to generate reduced form of cytochrome C. Then we have FADH2, which via the complex 2 that consists of the succinate dehydrogenase will catalyze oxidation of FADH2 to yield the reduced form of coenzyme Q. And then again, this reduced form again will be later on used by complex 3. So just remember, complex 1 is made of, consists of the activity of NADH dehydrogenase, while complex 2 consists of activity of succinate dehydrogenase. Then we have complex 3, which consists of cytochrome C, and this complex catalyzes oxidation of coenzyme Q. So this coenzyme Q will now undergo oxidation in order to yield a reduced form of cytochrome C. And then finally, complex 4 catalyzes oxidation of cytochrome C in order to transfer electron to oxygen and yield H2O. And every one of these reactions are releasing energy that are used to pump protons or H+ from mitochondrial matrix to the mitochondrial inner membrane space. So now we have an electrochemical gradient that has built up in the intermembrane space. And so that's where we need the electrochemical gradient for ATP synthase in order to function properly. So now let's go over the function of ATP synthase. So there we go. Here I have a drawing of ATP synthase for you. And so basically ATP synthase, or referred sometimes to as complex 5, it's a membrane spanning enzyme that catalyzes phosphorylation of ADP into ATP in a reaction that is carried out by the electrochemical energy from electrochemical gradient from the H+ that has built up in the intermembrane space. And so I'd like you to pay attention to the structure of the ATP synthase. It has an F0 unit, which is a hydrophobic segment that has an ion channel that allows electron to move from the intermembrane space to the matrix. And here, for simplicity, I've shown that the H+ is actually running through the pore, but it's actually from the side that H+ will enter from the intermembrane space into the matrix. And by doing so, it will cause this energy from movement of H+ will cause rotation of the F0 subunit. As a consequence of which, rotation of F0 subunit will also cause rotation of the subunit shown here and conformational changes in the alpha and beta subunit pairs of the F1 subunit. F1 unit of the ATP synthase. And now these conformational changes allows ADP and phosphate to enter through these complexes. They're kind of getting squeezed together by all these conformational changes, and they will yield an adenine triphosphate. So adenine diphosphate plus a phosphate group will yield ATP. Now every NADH we just discussed, we had 3 H+ moved, so will yield approximately 3 ATP, while every FADH2 yields approximately how much? 2 ATP. Alright, so that's about it. We're done with the electron transport chain. Next, I'd like to review the regulation of oxidative phosphorylation, and then finally, overview of aerobic respiration will help us conclude the aerobic respiration. So for the regulation of oxidative phosphorylation, again, what molecules will enhance oxidative phosphorylation? And it's easy to know because ADP means that we don't have enough energy. So if there is high concentration of ADP, therefore there would be increased activity of the ATP synthase enzyme. Likewise, if the ATP to ADP phosphate ratio, if this is too low, then we know that we don't have enough ATP, or there is too much ADP that is going on. Again, there would be increased activity of ATP synthase in order to increase the ATP production. And then finally, one other determinant of the ATP synthase activity is availability of oxygen. Under ischemic conditions, in other words, once there is oxygen deprivation, electron transfer to oxygen as well as pumping of the protons are ceased. And so under these circumstances, ATP synthase could operate actually in reverse, and hydrolyze ATP. And that would be very catastrophic because we are already not having enough oxygen. If we destroy the remaining ATP, it could severely damage the cells. And so in order to avoid the reverse operation of ATP synthase, there is this protein called IF1 protein inhibitor, which will lock the ATP synthase in place. And this IF1 protein becomes active once there is a low pH. So once there is not enough oxygen, too much lactic acid is produced, and there is low pH, IF1 will become activated and block ATP synthase. Once there is oxygen availability is restored and pH raises back to normal, then IF1 will change into its inactive conformation and let go of ATP synthase and allow it to function again. So again, IF1 prevents ATP synthase from functioning in reverse once there is low oxygen. So again, ADP, high ADP, low ATP, or ischemic conditions that can activate IF1, these are all helping to regulate the activity of ATP synthase in oxidative phosphorylation. Now, as for the overview of aerobic respiration, we've already discussed that we got a total of 4 ATP via substrate level phosphorylation plus 10 NADH plus 2 FADH2. And if you don't remember how we got all those, here I outlined it again. Glucose conversion to pyruvate gave us 2 NADH and 2 ATP. Pyruvate to acetyl coenzyme A gave us 2 NADH. And then finally, citric acid cycle gave us 3 NADH, 1 FADH, and 1 ATP. But since we had 2 molecules of pyruvate from here on, everything is multiplied by 2. And if you add up all these numbers, we get 4 ATP, 10 NADH, and 2 FADH2. Now, the question that I have for you is, assuming that 1 NADH gives us 3 ATP and 1 FADH2 gives us 2 ATP in the electron transport chain, how many ATP do we get from breakdown of 1 glucose molecule? So 10 times 3 equals 30. 2 times 2 is equal 4. So 4 plus 30 plus 4 would give us 38 ATP. And then you might have noticed that some sources say 36 to 38 ATP is produced from 1 molecule of glucose. Can you tell me where this number 36 is coming from? And so the number 38 is seen in bacteria because they don't have any membrane-bound organelles, versus in eukaryotes, since the 2 NADH that are produced by glycolysis is in the cytoplasm, and now it has to enter the mitochondria in order to undergo the electron transport chain. And this transfer of NADH from cytoplasm to mitochondria in the eukaryotes can cost energy, 1 ATP per NADH molecule. So that's why eukaryotes are making 36 ATP compared to prokaryotes, which are making 38 ATP from 1 glucose molecule. And if you're curious how the NADH will be transferred from cytoplasm to the mitochondria, here I'm showing you the Mullet shuttle. So it's actually being transferred via the Mullet shuttle. And so you can see here, by converting oxaloacetate into mullet, and NAD plus will be formed. Mullet can cross the membrane, and now it will be reverted back into oxaloacetate in order to yield back the NADH. So here we have transferred an NADH molecule from cytosol to the mitochondria, but this whole process will use 1 ATP. And so since we have 2 NADH molecule that we need to transport, therefore 2 ATP will be used up. So instead of 38 ATP that we've seen produced from 1 glucose molecule in prokaryotes, since 2 ATP had to be used up for transferring NADH from cytosol to mitochondrial matrix in the eukaryotes, eukaryotes are producing ATP. Now, one final point about aerobic respiration is that all these values about 1 NADH giving 3 ATP and 1 FADH giving 2 ATP, these were obtained with different lab experiments. And some of the newer lab experiments are coming to the conclusion that NADH is actually giving us 2.5 ATP and FADH2 is giving us 1.5 ATP. And that's the reason that some of the new sources are telling us that one glucose molecule yields 30 to 32 ATP during oxidative phosphorylation. And here I'm going to calculate the values with you just to show you how. So if we have 2.5 ATP produced per NADH, 2 times 2.5 gives us 5 ATP. But then this NADH from glycolysis is inside the cytoplasm. So if you're talking about eukaryotes, it takes another 2 ATP to move this NADH. So therefore the net yield could be 3 if you're talking about eukaryotes. Then we have other 2 ATP from substrate level phosphorylation. Pyruvate oxidation gives 2 NADH. So from here on, I'm multiplying NADH numbers by 2.5. So 2 times 2.5 gives us 5. 6 times 2.5 gives us 15. 2 times 1.5 gives us 3. So 1.5 ATP for FADH. 2 times 1.5 gives us 3. And then we have another 2 ATP here. Now let's add up all these numbers. 5, 20, 30. So 30 ATP in eukaryotes, or 32 ATP is yielded in prokaryotes. And that's about it. Now we are done with the next phase of this lecture, which was oxidative phosphorylation. So far we've explained glycolysis and aerobic oxidation. The final topic for the first half of this lecture that we still have to discuss is anaerobic reduction or fermentation. And here we've shown you that if there is oxygen, the pyruvate will undergo aerobic oxidation. Now what if there is not enough oxygen? So therefore the pyruvate molecule has undergo anaerobic reduction through the process called fermentation. All right, now let's discuss fermentation. There we go. So tell me again, remind me, what was the purpose of fermentation? So during the glycolysis, all the NAD plus are used to be converted into NADH in order to generate ATP. But the problem is that if we run short of NAD plus, glycolysis cannot carry out anymore. So therefore, by undergoing fermentation, the NADH can be converted back to NAD plus in order to allow the glycolysis to go through. And so the reactions that are going on, if you're talking about animals or bacteria, pyruvate can be converted into lactate through the process called lactic acid fermentation. And actually I have the second reaction right here. Just pay attention to the second reaction right here. You can see that the pyruvate via the lactate dehydrogenase enzyme produces lactate. Through this process, NAD plus is regenerated. So that's lactic acid fermentation that is seen in animals and bacteria. And then finally, we have the ethanol or alcohol fermentation, which is seen in yeast and some bacteria, where pyruvate, with the help of pyruvate decarboxylase, is converted into acetaldehyde, this process releasing a CO2, as a consequence of which now we have a 2-carbon molecule. And then finally, alcohol dehydrogenase will help produce an NAD plus from NADH, and the final product is ethanol, which is also a 2-carbon molecule. Now, before I conclude the first half of this lecture, I'd like to give you a real-life example of how wine is actually being produced. So if you get a bunch of grape, squish them and place them inside a glass container. So here is the squished grape, and it has a lot of yeast that is naturally present on the grape. And now I'd like you to place an airlock on top of here. So we have some water on here. Air can go out but cannot go in. What happens is that it will produce an anaerobic condition. So all these yeast cells that are inside the container need to produce ATP. So they will have to undergo glycolysis, but then all the NAD plus is used up. In order to produce more NAD plus, they will have to undergo ethanol or alcohol fermentation. And so NADH will be regenerated, but then there will be also increased ethanol production and increased CO2. And that's why that you will see that bubbles keep coming out of the container. It's because the CO2 is being released throughout this fermentation reaction in order for glycolysis to carry out. And then eventually ethanol concentration will increase to a point that it will kill all the yeast inside the container, and so the reaction will finally stop at that stage. So basically that's about it. First half of this lecture is done. We've covered glycolysis, fermentation, citric acid cycle, and electron transport chain. In our next lecture, we will go and review the pentose phosphate pathway, gluconeogenesis, glycogenesis. Let's not confuse glycogenesis from glycolysis. Glycolysis is breakdown of glucose. Glycogenesis is production of glycogen. And in regulation of these pathways, again, we discuss. And then finally, we will conclude the second half of this lecture with discussing the metabolism of fatty acids and proteins, and then giving you some practice questions to prepare you for your MCAT. Thank you all for listening, and I will see you for the second half of this lecture.